Very good evening to all of you. I welcome you all to today's Wednesday webinar on genomics. I'll request our President Dr. Shiva Gulati to introduce the speaker and the experts for today's session. Go to you, ma'am.
Yeah, thanks, Asmita. Yeah, so today we have Dr. Shiva Prakash Ramalingam, who's a principal scientist in the Division of Genomics and Molecular Medicine at CSIR Institute of Genomics and Integrated Biology, New Delhi, who has co-authored more than 50 research articles in peer-reviewed journals. And the research focus of his has been on the development of cell and gene therapy for hematological diseases. So today we will be discussing basically what are the new recent advances which have been there in the, the management of the newer therapies which are coming, the emerging therapies which are there for the genetic diseases. And this is very important and it's very upcoming, and it has revolutionized many conditions, which were not having amenable to therapies are now coming up. And the only thing is many of them have prohibitively expensive. So we're looking for more, having more research in India so that you develop more molecules which are more accessible and approachable. So over to you, Dr. Shiva Prakash, and we will be having Dr. Vinod Skaria, who you all know very well as an expert. Dr. Madhulika Kavra is unable to join because she's not feeling well. We all will be Dr. Vinod, Dr. Mahesh, myself will discuss the, about how the emerging therapeutics or genetics diseases there. Over to you, Dr. Shiva Prakash.
Thank you, madam. Thank you, Vinod. Next, maybe for 45 minutes, I'll be talking about emerging therapeutics for genetic diseases. So this is the agenda of my talk. So I will just start with the gene therapy, the basic, what is the definition for gene therapy. And then I will also talk about ex vivo and in vivo gene therapy and their limitations in the treatment of genetic diseases. And then in the second part, I will be talking on genome editing. What is genome editing and their clinical applications. And then finally, I will touch upon what we are doing at IGAB in my lab for hemoglobinopathies, especially for sickle cell anemia and thalassemia. Just giving an example for this treatment approach. So what is gene therapy? This is the simple definition for gene therapy. Gene therapy is nothing but introduction of therapeutic or healthy gene into the patients using a viral vectors to treat genetic disorders. So say example, we can give many genetic disorders, sickle cell anemia, thalassemia, or hemophilia, or neurological diseases like SMA, or lysosomal storage diseases like Gaucher's, Pompe, and other things. So those are all the genetic disorders. There is a defective gene, which is either there is a mutation or point mutations or deletion. So in those cases, what you can do is you take a wild type or the therapeutic gene, package them into a viral vector that is being used as a vehicle to deliver your therapeutic gene into the cell type. Okay, there are two types of cell and gene therapies. One is the in vivo gene therapy and another one is the ex vivo gene therapy. So if you see in the left, I have shown the direct delivery of therapeutic gene, which is the, just 1 minute. Yeah, so this is the direct gene delivery. So here mostly adeno-associated viral vectors has been used. So I'll give you an example in my next slide, which is been FDA approved for SMN. So you take the therapeutic gene, which can be in the case of sickle cell or thalassemia, it's a hemoglobin. Package them into a suitable adeno-associated viral vector. So there are in adeno-associated viral vector, there are different serotypes are there. It's 1 to 11. So the serotypes is nothing but in simple terms. Say example, adeno-associated virus 6, AAV6 type, which mainly infects the hematopoietic stem cells, hepatocytes, all those things. So AAV9 mostly infects the neurons. Okay, so it's like a cell type specific. So you directly inject your viral vector, adeno-associated viral vector, intravenously into the human body. So that it goes into the respective cell and then it transcribes and then it produces the protein and it expresses. So that is the in vivo gene therapy. So you are directly intravenously injecting into the patient without taking out the cells. But on the other side, the second approach is called as the ex vivo approach. So there you take out the harvest the stem cells, say example in the case of sickle cell, thalassemia or hemophilia. In those cases, you take out the patient blood through the apheresis method and then purify the respective cell type. Say in the case of beta hemoglobinopathies, you take out the CD34 hematopoietic stem progenitor cells. Okay. And then package the therapeutic gene using the lentiviral vector approach and then deliver it into the respective cell type, CD34 or other cell types. Okay. So this happens in the GMP grade facility, GMP facility, because this has to be in a clean room facility. So then after the genetic modification, this can be put it back into the patient. So this is the ex vivo approach. So I'll give you just a couple of examples. So one is the very well-known example is the spinal muscular atrophy. Okay. So in last year, FDA has been approved this therapy. This is the AAV9 based therapy. Okay. So what they have done is they have packaged this SMN1 gene, survival motor neuron gene, in a AAV9 vector. So which specifically transduce the motor neurons. That is the nature of this AAV serotypes. Okay. So as I told in my earlier slide, the AAV6 mostly infects the hematopoietic stem progenitor cells. So similarly, the AAV9, which mostly infects the motor neurons. So here it is like a in vivo gene therapy. So what they have done is they have taken the SMN1 gene, packaged into the AAV9 vector, and then directly intravenously they have injected into the patient. Okay. So so then eventually the patient will the AAV goes into the motor neuron cells, it transduces, and then the protein gets the DNA gets transcribed into mRNA and then translated into the protein. So this is a one-time intravenous injection. So the transduction of motor neurons likely results in improved motor neuron development, function, and survival, with the potential for near normal attainment of the motor milestones. Okay, so this has been very successful gene therapy approach. So the second gene therapy, which has been also recently approved, this is from the Blueboard Bio. It's called as Lentiglobin. So this is mainly approved for the beta thalassemia. Okay. So here they have used the lentiviral particle. There in the SMA, it's a adeno-associated viral vector. So here it is another viral vector which is called as lentiviral vectors. Okay, so which transfects the primary cells, especially the progenitor cells. So here they have packaged the beta globin gene with appropriate promoter and the signaling sequences, the locus control region sequences into the lentiviral particle. And then in ex vivo, they have taken out the patient's CD34 hematopoietic stem progenitor cells in the GMP condition. They have transduced these lentiviral particles into the CD34 cells, and then after that they have transfused these cells, the genetically engineered cells, back into the patients. So here the major problem in beta thalassemia is the globin chain imbalance. I will talk more about it. So if you see here in the first vector BB309, because of the beta globin mutation, there is a excess production of alpha chain, less production of the beta chain. But once after transducing these lentiviral particles into the CD34 cells, there is a higher expression of the beta globin. So that's where these vectors used as a vehicle, okay, into for transferring your therapeutic gene into a specific cell type. Okay, but there are some limitations with these the gene therapy approach. So it has been successful, but there are some limitations which has to be addressed. There are two major limitations. One is there is a very low rate of homologous recombination, especially in lentiviral approach because this is the lentiviral approach. It goes and randomly integrate into the genome. Okay, So it is not specific to the particular chromosome or at the specific locus. Okay, so this it goes and randomly integrates in the genome. So the randomly integrates means there is a low rate of homologous recombination in the mammalian system. So in mammalian system, the homologous recombination efficiency is very, very low. It happens 1 in 1 million cells. Okay, so that's where the major problem. It's randomly integrates, and then the random integration leads to sometimes it may go and integrate into the oncogene sites, oncogene promoter site, and then the oncogene may get activated. So eventually the patients may develop a leukemia. So that is one major limitation with the lentiviral based approach. Okay, so one of the best example for this random integration is the SCID therapy. Okay, so you might have seen read in the textbooks and the newspapers this SCID, the severe combined immunodeficiency syndrome, which is also called as bubble syndrome, which is a genetic disorder in which the patient lacks the type of immune cells. This is because of the mutation in IL-2 receptor gamma gene. So this happened in early 2000, this gene therapy. Okay, what they have done is they have packaged this IL-2 receptor gamma gene in a retroviral vector, okay, which is another type of the HIV based vector. So they packaged into the retroviral vector and then they delivered it into the 11 patients. Okay, what happened? Unfortunately, as I told earlier, these viral particles went and integrated into a oncogene site, and then the oncogene promoter, oncogene promoter site, and then it got activated. This the viral particles get activated, and then a patient got eventually developed a leukemia, and then he died. So this is one of the major limitations. Because of this miracle gene therapy got halted in early 2000. Okay, so that is one of the major setback in 20 years back. But the gene therapy field has been moved from there, and now from second generation to third generation lentiviral particles has been used in all the recent clinical trials and the approved ones. So which seems to be very, very safe. If you see in the CAR T cell therapy, which is not genetic, but it is for the leukemias, especially the ALL and DLBCL patients. So it's been injected to more than 10,000 patients. It's been followed up for more than 5-6 years. It seems to be very, very safe. Okay, but still there is some concern among the scientific field and the clinicians, the random integration. So how to address this random integration? Okay, so that's where the question we and others ask is, can we precisely edit a gene? Say example, in the case of, as I told, sickle cell anemia or thalassemia, or in the case of spinal muscular atrophy, SMA. So can we edit the particular mutation in the SMN1 gene? Or in the case of Gaucher's disease, can be able to edit the L44P mutation, which is also involved in the Parkinson's L44P, the heterozygous one. Okay, so that's where the genome editing comes. So I'll give you just a brief introduction about the genome editing. This is the definition. Genome editing is the process of introducing specific modifications in the endogenous genomic DNA, including it can be deletions, point mutations, inversions, or translocations by targeted double-stranded break. Okay, so you can by using genome editing, it makes a targeted double-stranded break. So the targeted double-stranded break leads to the different kind of, it creates different kind of mutations. Okay, either it can be point mutation, inversion, or translocations. By using genome editing and combining with the specific donor molecule, we can enhance the homology directed repair. As I told in earlier, if you are using lentiviral based approach, the integration, the targeted integration happens only in 1 million cells. Okay, but if you are making a targeted double-stranded break, the homology directed repair efficiency goes up several thousand-fold. Okay, that's where the genome editing makes the lot of difference. Okay, so if you make a targeted double-stranded break using gene editing nucleases, it makes a cut at the target place, and then using the donor DNA by homology directed repair, you can able to correct the particular mutation. Okay, so why genome editing? As I told, one basic application, not only basic, it's a clinical application is you can use for the genetic correction of a particular mutation, the particular genetic mutation. The second is also it can be used to understand the basic biology. Okay, in our human genome, we have around 20,000 genes. But we know only the 6,000 genes function. Okay, so the remaining 14,000 genes, and there are some long coding RNAs, microRNAs and other things. We don't know those functions. So in order to study the basic biology, you can go on, you can take a cell type, specific cell type, do a knockout using the gene editing nucleases with that specific gene or the LNC RNA or miRNA, and then study the basic biology, whether the gene is essential for cell growth and development, and then it involves in what kind of pathway, or how it regulates the downstream and upstream process, upstream genes. Okay, so there are 3 gene editing nucleases has been so far reported. The first generation gene editing nucleases is zinc finger nucleases. Zinc finger is the DNA binding protein. We all know that, which is the most abundant DNA binding protein in the eukaryotic system. What this scientists have done is, they have taken the zinc finger DNA binding protein and then fused a type 2 restriction enzyme, which is called the FOC1 restriction enzyme, and then created a zinc finger nuclease. So this was done by Professor Chandrasekharan's lab at Johns Hopkins University in way back in 1996-97. So it was shown that in many cell types, even including the primary cells and other model organisms like Drosophila, C. elegans, and other things, if you make a zinc finger nucleases for targeting any gene, deliver it through either it can be by chemical mediated method or the lentiviral or electroporation method. It goes inside the cell and then it binds at the specific locus, and then the FOC1 restriction enzyme dimerizes and it makes the double stranded break. So once the double stranded break happens, by using a homology directed repair, the gene can be corrected. So this is the first generation gene editing nucleases. And then subsequently, there is another gene editing nucleases was reported, which is called as TALENs. Okay, it's a transcription activator like effector nucleases. In the case of zinc finger nuclease, each finger recognizes 3 base pair. Okay, so you put together 5 zinc fingers, which can recognize 5 into 3, 15 DNA in the genome. Okay, so since FOC1 has to dimerize, you need to have two molecules, okay, of zinc fingers. Okay, so then only you have to have it has to dimerize, and then it makes the targeted double stranded break. So there is a left zinc finger and there is a right zinc finger. Okay, so the left zinc finger, as I told, if you combine 5 zinc finger molecules, it targets the 15 base pair, and then the right zinc finger also, you can able to make 5 zinc finger molecules by combining 5 zinc fingers. So that can also target 15 nucleotides. Okay, so the 30 nucleotides of zinc finger target region, at a stretch, it may not be present elsewhere in the genome. So that's where the specificity comes into the picture. Okay, so similarly, the TALENs, the transcription activator effector nucleases. So in the zinc finger nucleases, each zinc finger binds 3 bases, but here the DNA binding domain is the tail. It's a transcription activator like effector protein. Okay, this was isolated from a Xanthomonas plant bacteria. So scientists from Max Planck Institute, Dr. John Poch, he's a plant pathologist. When he was working in this bacteria, he identified this TAL transcription factor. So what scientists, since the zinc finger nuclease was already there, so what the scientists have done is, so they have combined the different tail domains and then fused it with the FOC1 restriction enzyme, the type 2 restriction enzyme, and created a TALENs. Okay, it's a transcription activator like effector nucleases. Okay, so the nuclease is same for zinc finger as well as the TALENs. Okay, the only difference between zinc finger and TALENs is zinc finger binds 3 nucleotides, but the tail domain which binds, it recognizes only one DNA, only one base pair. Okay, so here also you can combine 14 to 15 tail DNA binding domain. And then at the 3-terminal end, we can fuse the FOC1 nuclease, and then we can create left talon and right talon. So if you deliver it into the cell, it goes and binds to the respective site, and then it makes the targeted double-stranded break. Okay. So the third one is the CRISPR-Cas9, which is also the third generation gene editing nucleases. So this is something like a restriction enzyme. Okay, so like as bacteria also get infected with viruses, right? In order to fight off the infection, so bacteria also has evolved a defense mechanism. One of the mechanism is the restriction enzyme based approach. The second one is there are many things in among those. One of them is the CRISPR-Cas9. Okay, so the CRISPR-Cas9 is RNA and a protein system. Okay, but in the case of zinc finger and talons is completely a protein based approach. But here you have a synthetic guide RNA which has a specificity towards the your target molecule, and then the Cas9 is a nuclease, which is something similar to FOC1 nuclease. Okay, so so here what happens is the RNA molecule which binds to the target sequence, once that it binds, there is a PAM, protospacer adjacent motif sequence in the downstream of your the guide RNA sequence. So the Cas9 which recognizes the PAM molecule, and then it makes the targeted double-stranded break. Okay. So, so all these nucleases, zinc finger nuclease or talons or CRISPR, it does the same job, which is nothing but a targeted double-stranded break. Okay. So among these three systems, the CRISPR-Cas9 is much more simpler than the zinc finger nuclease and talons. So that's where it has transformed the field, actually, the biology, not only biomedical science, it has transformed it into the livestock and the agriculture biotechnology. Okay, so so targeting multiple genes is much more achievable using the Cas9 multiplex genome editing, because you can target multi genes using the same Cas9 molecule. Okay, but in the case of zinc finger and talons, you have to design a target for each domain, each target region. Okay, so that takes time, and then you need to have a protein engineering expertise, little bit of protein engineering expertise. So that's why the CRISPR-Cas9 approach is, in addition to that, it is also a much cheaper, because we need to have synthesized the 20 nucleotide sequence. And then it can be simply cloned into a vector which can be generated within a couple of weeks. Okay, so what is the application of this gene editing nucleases? Okay, so if you consider this is a cellular genome, okay, so if you want to make a particular mutation, or if you want to correct a particular gene, you take any one of the gene editing nucleases, either it can be zinc finger, talons, or CRISPR. You simply deliver it into the genome of your interest, or the cell type. So it goes directly into the cell, and it searches and it binds at the target locus. Okay, and then once it binds, it gets translated, it transcribed and translated into the protein, and it makes the targeted double-stranded break at the target locus in the human genome. Okay, once it makes the targeted double-stranded break, cut happens. Then the cells will take care about the repair mechanism, because it can't be once it cut has happened, it has to get repaired. Okay, so the repair mechanism, there are two types of repair mechanisms. One is the non-homologous enjoining based repair mechanism. Another one is the homology directed repair mechanism. Okay, if you don't provide any donor DNA molecule in your zinc finger nuclease, talons, or CRISPR delivery system, so it goes and cuts, and then by non-homologous enjoining based genetic disruption happens. So during this non-homologous enjoining, there may be a small deletion, insertion, or base pair substitution has happens in the target region. Okay, basically it makes a knockout. Okay, so the gene it may put premature stop codon, or there may be a long deletion, few base pair deletion or long deletion. There may be a 5-6 base pair deletion, or in some cells it may be a 100 base pair, 200 base pair deletion also may happen. Okay, so that is nothing but a gene knockout. Okay, so the second approach is genetic correction. So along with your gene editing nuclease, if you provide a donor DNA molecule with appropriate homology arm. So homology arm is nothing but where the cut has cut site is there. So you take maybe around 500 to 1 kb region. You use that as a upstream 5' homology arm to your correction site, and then take downstream. There may be a 1 kb, 500 to 1 kb region, and then make a donor DNA template. Okay, so that you deliver it along with your gene editing nuclease. Once that cut happens, it uses the cells its own repair mechanism. It uses this as a template, okay, for genetic repair mechanism. Okay, so then since it has the homology arm on both the sides, so the repair happens, and then it gets inserted at the cut site. So that's where the genetic correction happens. Okay, so the third approach is you can also overexpress a gene of interest. Okay, so this can be used for enzyme replacement therapy. Okay, you don't need to correct a particular mutation, but in the human genome, there are certain sites or safe harbor. It's called a safe harbor locus. Okay, say example one is the CCR5 region. CCR5 is nothing but a chemokine receptor. Okay, so where HIV uses to enter into the T cells. Okay, so we all know that there are some tiny group of population across the world. They are having 32 base pair deletion in the CCR5 gene. Okay, so that those people are either it can be a biallelic or monoallelic deletion. So those who have biallelic deletion, the 32 base pair deletion, they are permanently resistant to HIV. So even though they have these deletions, they live very normal life, okay, without having any cell growth and development effect. Okay, so that region, the CCR5 region, can be used as a safe harbor locus to insert a foreign gene. Okay, so if you want to do a enzyme replacement therapy for, say, example, lysosomal storage disease, you just take the gene with the appropriate promoter sequence, promoter and regulatory sequence, insert that gene into a CCR5 locus, or there is another locus called AAVS1 locus. Okay, so that locus also can be used in the human genome to insert the specific gene. So this is for the targeted insertion, not like a lentiviral based approach. It randomly integrates into the genome. So the take home message is the homologous recombination is increased. You can 1,000-fold more efficient during the homology directed repair. Okay, so that is the take home message. Okay, so with this, I will just give you a specific example. Our lab is working on genetic disease, which is nothing but a beta hemoglobinopathies, which includes the sickle cell anemia and thalassemia. The question we ask is, can we cure using this new technology, which is nothing but the genome editing approach? Okay, so can we cure this genetic disease? Because right now there is no cure available except the bone marrow transplantation. But recently, there's just last year, the lentiviral based approach was approved, FDA approved only for beta thalassemia, but there is no therapy still available commercially for sickle cell disease. Okay, so the question we ask, can we cure beta hemoglobin diseases using gene and cell therapy approach, or nothing but genome editing approach? Okay, so just to give a brief introduction about the sickle cell and thalassemia. So in sickle cell, you know that there is a point mutation, which is at the amino acid 6th position, glutamic acid to valine, which alters the shape of the blood so that the oxygen carrying capacity gets hampered due to the sickle shape. Okay, so on the other side, the gene is the same in the case of beta thalassemia, but the mutation is, there are 200 different mutations. The mutations are spread across the gene. Okay, because of this mutation in the hemoglobin gene, the adult hemoglobin, so there is an excess production of the alpha globin. Okay, so this, because of this chain imbalance, okay, so that leads to reduced erythropoiesis. Okay, that's where these beta thalassemia patients have to go for regular blood transfusion once in 2 weeks or 3 weeks. It depends on the severity of the symptoms. Okay, so these two diseases are very, very prevalent across the world, even in our part of the country, our part of the world, where each year almost like 20 to 30,000 children are born with the sickle cell, and almost 10,000 children are born with the thalassemia. Okay, so as I told, there is no cure except the bone marrow transplantation. There are a couple of drugs are available like hydroxyurea, voxelator, L-glutamine, and other things. But if you see, this is for sickle cell disease, but in the thalassemia, only blood transfusion is being practiced for several decades, but there is no cure except the bone marrow transplantation. But you know that bone marrow transplantation has several disadvantages in like GVHD, and there is low lack of donors and graft rejection, transplant related complications and other things. Okay, so that's where the gene and cell therapy plays a major role. So so I'll just briefly touch upon how the hemoglobin gene is being developmentally regulated. As we all know that there are 5 hemoglobin genes present in our human genome. So each gene is developmentally regulated. Okay, so at the embryonic stage, embryonic hemoglobin is expressed, and then the fetal during the fetal stage, the embryonic hemoglobin gets silenced and the fetal hemoglobin expressed. And then the second switch happens during the pregnancy, after the during the pregnancy, that is the fetal hemoglobin. And then after the birth, both embryonic and fetal hemoglobin gets silenced, and then the adult hemoglobin starts expressing after the 6 months of the age. Okay, but there are some individuals, even at the adult stage, they express the fetal hemoglobin. Okay, so throughout their life. Why? Because there are some mutations, natural mutations, which is present in the promoter region of the fetal hemoglobin. Okay, these are all the point mutations. Or on the other side, there is a long deletion. Okay, so there is a complete deletion of the adult hemoglobin or in the mutant hemoglobin gene in the sickle cell or thalassemia patients. So those those individuals in the one allele, they have the this mutation. The second allele, they have the long deletion. Okay, so because of that, they express the high level of fetal hemoglobin. So they live long life without having any clinical abnormalities. So this is called as hereditary persistence of fetal hemoglobin. Okay, so either there is a point mutations in the promoter region of the fetal hemoglobin, where there are some negative regulators which binds. So because of the mutation, the negative regulators can't able to bind at the promoter region. So that's where the fetal hemoglobin expression is continued into the adult stage instead of the adult hemoglobin. Okay, so the hereditary persistence of fetal hemoglobin, which is the natural way to enhance the fetal hemoglobin expression. Okay, so the clinical symptoms of sickle cell disease and thalassemia can be alleviated with the co-inheritance of the HPFH genotype. Okay, so you no need to have 100% fetal hemoglobin to take care about the sickle cell, to alleviate the symptoms of sickle cell or thalassemia. So even around 20 to 30% is more than sufficient. The fetal hemoglobin level is more than sufficient to take care about the disease pathology. Okay, so the reactivation of fetal hemoglobin has a potential definite solution for both sickle cell and thalassemia. So these are all the different mutations. I'm not going into the details. Okay, so there are multiple approaches which can be used for this disease. One is, as I told, you can use the gene editing nuclease approach. Using the homology directed repair, you can correct the particular mutation. In the case of sickle cell disease, it's a single point mutation. It is very straightforward. You take out the hematopoietic stem progenitor cells, that is the CD34 cells, and then you just do the genome editing approach and then correct the particular mutation. Okay, but in the case of sickle thalassemia, there are 200 different mutations. It will be very cumbersome to develop a strategy for each and every mutation. Okay, so that's where you need to have a strategy which is independent of your gene correction. Okay, so that's where if you can able to reactivate the fetal hemoglobin using the HPFH approach, that will be a one-stop solution for both sickle cell and thalassemia. Okay, so that is where the approach we and others are doing. So instead of we are not doing any genetic correction, but instead of that, we are trying to create the naturally occurring mutation in the patient cells, and then so that we can increase the fetal hemoglobin levels in the patient hematopoietic stem progenitor cell derived erythroid cells, so that the patients will reverse the disease phenotype. Okay, so this is the hypothesis of our study. So if you see here in the left, this is the disease condition where there is a mutation, sickle cell mutation or thalassemia mutation. Those people, these patients have low level of fetal hemoglobin. That's why they have the disease phenotype. Okay, so by using genome editing, can we, if we can delete the mutant gene along with the negative regulators region, which is around 50 kb region, which is nothing but hereditary persistence of fetal hemoglobin 3. So there are some Indian individuals, you can't call them as a patients, because these are all normal individuals. They have this long 50 kb deletion. Okay, so they have very high level of fetal hemoglobin, around 30 to 35% of fetal hemoglobin across the generations. Okay, without having any clinical abnormalities. So that gives some clue for us, so that we can able to use genome editing to recapitulate those naturally occurring mutation in patient cells. Okay, so if you create those mutation, then you can able to remove the negative regulator region and the sickle beta hemoglobin gene region, so that you can reactivate the fetal hemoglobin, so that you can reduce the disease phenotype. Okay, so this is the strategy we have followed. So we have taken the sickle cell or thalassemia patient CD34 hematopoietic stem cells from the PBMCs, and then used the genome editing approach. And then we have, before doing the in vivo experiment, we have differentiated them into erythroid cells, because the CD34 cells are the progenitor cells. Okay, the hemoglobin is expressed in the red blood cells. Okay, enucleated red blood cells. So you need to differentiate them into red blood cells. So that's what we have done at the in vitro stage. We have then subsequently did the functional characterization using different molecular techniques like flow cytometry, Western blot, and HPLC. So we have seen that increased level of fetal hemoglobin, and then we have taken these cells, injected into the immunocompromised mice to show that after genome editing, whether these cells can engraft in the immunocompromised mice, whether these cells can go into the bone marrow and different organs, mainly in the bone marrow. Okay, so that is what we have done. So so here after genome editing, so we have done functional characterization in patient cells. So one of the important experiment is the HPLC, high performance liquid chromatography. So to show that after genome editing, after making the 50 kb deletion, recapitulation of the HPFH, beneficial HPFH3 mutation, whether the fetal hemoglobin levels has gone up or not. So we have taken three sickle cell patients, 3 different sickle cell patients, and then it did the genome editing, and then differentiated these cells into erythroid cells, and then did the HPLC analysis for different proteins, fetal hemoglobin, adult hemoglobin, and the sickle hemoglobin. So you can see here, this is the fetal hemoglobin for the sickle cell patients. So this is the untransfected one, and this is the mock, which has only the Cas9 protein, which doesn't have the sgRNA, but this HPFH3, which has both sgRNA and the Cas9 molecule. So when makes the beneficial mutation, you can see here from this from 10%, it has gone up to almost like 30 to 35% of the fetal hemoglobin in all 3 patients. Okay, so that shows that, so if you increase the fetal hemoglobin, the next question whether it will bring down the sickle level. Okay, so that is where you can see here, this is the morphology. So we have taken the genome edited RBCs, red blood cells, given a hypoxia treatment, and induced the sickling in these cells. And then we have checked using the microscope. So this is the unedited one. So you can see here, most of the cells are still maintaining the sickling phenotype. But in the corrected ones, you can see very few cells are showing the sickle. So this is the counting. So in sickle cell, it's in vitro condition, it's almost like 70 to 80% of the cells are showing the sickle phenotype. But after genome editing, so it has come down to almost like 10 to 20%. Okay, so this shows that you can use genome editing, reactivate the fetal hemoglobin, and then reverse the disease phenotype. So another thing we have done is the reactive oxygen species. So this is one of the hallmark of the sickle cell disease pathophysiology. So in this, we have tested two patients. Okay, so in both the cases, you can see here almost like 50% reduction in the reactive oxygen level, reactive oxygen species levels. So this is similarly, we have also done for taken 3 different beta thalassemia patients with 2 different mutations. And then we have seen the fetal hemoglobin reactivation almost like 25 to 30% using the reverse phase HPLC. And then subsequently, as I told, after genome editing, you need to make sure that these cells can able to engraft in the bone marrow of the before going into the clinical, before going into the patients. Okay. So what we have done is we have taken the immunocompromised mice, and then we did the tailwind injection of these edited cells, and then we followed it up, followed it for 16 weeks. Okay. So this is the long-term engraftment analysis. Okay. So in each 4 weeks, so we have collected the blood and did the FACS analysis to check the engraftment. Engraftment is nothing but whether these edited cells can home into the bone marrow and bone marrow, mainly bone marrow. And then we have also tested with the thymus, spleen, and the circulatory system of the blood. Okay. So we have, this is the approach we have taken. So we have taken the NBSGW mice, which is the immunocompromised mice, which doesn't have the active B cells, T cells, and NK cells. So we did the tailwind injection. So after that, after 16 weeks, so we have collected, as I told, we have collected the blood each week, and then each week you can see here there is a increased engraftment. So this is the unedited cells, and this is the HPFS3 edited cells. So editing didn't hamper the engraftment of the edited cells. Okay, so we have also tested the editing efficiency in these cells. So editing efficiency is around 57%, and then the bone marrow engraftment is almost like 50 to 60%. Okay, so what are the lineages? We have also tested the B cell, T cell, and myeloid lineage, so which is comparable to the control unedited ones. And then we have after that tested the F cell population in the mice cells. Okay, so F cells is nothing but these cells, the F cells are the cells which produces the fetal hemoglobin. Okay, so in unedited, the F cell population is only 5%, but in the edited cells population, which is almost like 35%, it's almost like 7-fold increase. So HPFS3 edited hematopoietic stem progenitor cells repopulate for long term and produces HbF positive cells in vivo. That is the conclusion of this experiment. Okay, so similarly, as I told earlier, so we have also done with the beta thalassemia patients. In beta thalassemia patients, one of the major disease hallmark is the reactive oxygen species, so which has been drastically reduced in 3 different patients, almost like 50% reduction in these 3 patients. So which shows that if you can able to reactivate the fetal hemoglobin using genome editing approach, you can reverse the disease phenotype. So we observed the reduced levels of reactive oxygen species in edited cells compared to the unedited cells. So this is the overall approach when it reaches to the, so we have shown, we have taken a genome editing approach, taken both sickle cell and thalassemia patients CD34 hematopoietic stem cells using genome editing, deleted a region in the beta globin cluster at chromosome 11, and then differentiated those cells into erythroid cells. At the in vitro, we have shown that there is a fetal hemoglobin reactivation, and then in vivo, we have injected these cells into immunocompromised mice, taken those mice cells, bone marrow cells from the mice after 16 weeks, and then we have done the fetal hemoglobin levels and then ROS levels. So that shows that you can use genome editing, reverse the disease phenotype. So when we reach the this is the overall approach for the ex vivo genome editing. So you take out the patient's hematopoietic stem cells from using the apheresis approach. First, you have to mobilize to empty the bone marrow. To empty the bone marrow, you have to give the busulfan treatment and then take out the cells, CD34 cells, and then use the CliniMax Prodigy system, and then using non-viral mediated genome editing tools like sgRNA and Cas9 to electroporate these sgRNA Cas9 into the CD34 cells. And then once the patient is ready with the conditioning, so you can inject these cells, because it's autologous cells. You are taking the patient's own cells, modifying and putting it back into the cells. There won't be any immune rejection. The patient will get eventually cured. So this is the overall schematics of the cell and gene therapy, genome editing based cell therapy for sickle cell and thalassemia patients. So with this, the key takeaways is, here we have shown that the forced long-range chromatin interaction can be used to reactivate the developmentally silenced gene. Our approach consistently produced higher fetal hemoglobin levels in the 3 cellular models that we have tested. So this is a one-stop approach. It's a non-homologous enjoining based approach, which is we no need to do the gene correction. So that you can avoid the beta globin gene correction. So it is very cumbersome to develop the strategy for all the 200 different mutations. So, and then we have recently also successfully tested the edited patient-derived HSPCs in vivo in preclinical mouse model for the engraftment potential. So with this, I will stop here. So with this work was mostly done by 2 very good graduate students, Sangam Goswami and Pragya Gupta. So this work also was done in close collaboration with Dr. Kaurav Karya, who is a bone marrow transplantation expert at Indraprasada Apollo Hospital. So where we have collected the patient samples for this work. So this work is could have not been done without collaboration with Dr. Kaurav Karya. So because this is 100% clinical aspect, so clinical collaboration is very, very important to use these technologies if you want to study this in patient-derived cells. Okay. So with this, I will stop here. So if there is any questions, I'll be happy to answer. Thank you.
Thank you, Dr. Shiva Prakash. It was an excellent talk. Dr. Vinod, maybe, you can take on from here, as because now it is in the third in the series of, webinars which we're having on this genomics. So you can just take it forward and how the pediatric neurologists who are listening to it can benefit. And it is a very good work, amazing work which you are doing, Dr. Shiva Prakash.
Thank you, madam.
Thank you. Thank you. Vinod, to you.
Yeah, thanks, Shiva. You pretty much made it very simplified so that everybody can understand what genome editing is all about. But I mean, having said that, it is not as simple as he said in the lab. Dr. Shiva knows that very well. Well, my question is also something that you also alluded to, like, for example, there are many conditions which are caused by gene mutations, which could be treated by gene therapy or genome editing, especially which present in as a neuronal phenotype. Because typically when we talk about neurological disorders or neuromuscular disorders, we typically think about diseases which are very unlikely to be treated because of the fact that they affect neurons primarily. So could you provide some insights into diseases which in the neuromuscular or neurological spectrum, which are likely to be treated or are in clinical trials in the recent years?
Yeah, so one of the example, thank you, Vinod. One of the example is which is already been approved for this AAV9 based thing for the some spinal muscular atrophy, right? SMA. So because it is not like the circulation, right? So you can't take out the cells like sickle cell or thalassemia, because it has to be, you have to do a directed delivery approach. So there are two things we can think of. One is the AAV based approach. The second one is the nanoparticle based delivery. Okay, so if you can able to package the gene of interest into the nanoparticles, or nanoparticles, so which can specifically targets the neurons. So then the gene correction can happen, so there is a two approach in nanoparticle based approach. One, you deliver the genome editing reagent as well as your the therapeutic gene, so that it cut happens at the neurons, whichever the respective neurons, and then by homology directed repair, it can get corrected. The second one is you simply just deliver the nanoparticle mediated delivery of the therapeutic gene. So it will stay as a episomally, like AAV, adeno associated viral vectors. So, but the long-term effect is still we don't know. Okay, so AAV is also a episomal one. It doesn't integrate into the genome. So, but still there is at least 5-6 years of data is available for hemophilia. So there is a long-term persistence is there. So there are two approaches. One is AAV based approach for the neuronal diseases, or the nanoparticle based delivery. So because it has to pass through directly, it has to go through the neurons.
May I ask one, Dr. Shiva, before Vinod
ma'am.
asks, what about genome editing in iPSCs?
Yeah, no, that it can be used as a model system right now. So there are some clinical trials has been started in Japan for Parkinson's disease, I think. There is a trial going on at Riken in Japan for Parkinson's, and even for Alzheimer's, I think. So they have done some in vitro and in vivo work, and then they have also done some work in dogs and monkeys as well. In iPSC, this is based on iPSC based one approach only. So you need to have a GMP grade iPSCs. So then differentiate them into respective neurons, either dopaminergic neurons or any other respective neurons, and then using a specific delivery system. So they have used some specific needles injected directly into the specific region into the brain. Okay, so that also can be possible. Yes. Because in iPSCs, just I want to add up, madam. So from iPSCs, the differentiation in differentiating into neurons is very, very well worked out. Okay, so the getting the terminally differentiated neurons is almost like very well standardized protocols are currently available. Not like the CD34 hematopoietic stem progenitor cells. We can't able to achieve the fully matured CD34 hematopoietic stem cells. But in neurons, yes, it is possible to achieve terminally differentiated neurons. Yes.
Even for muscles, because VTA11 has got an orphan drug approval, which is an iPSC after genome editing for Duchenne muscle dystrophy in the US.
Right.
And and also for just reminding our friends who are there, obviously you have that Zolgensma, which has been there, which was been approved in May 2019, but we have now this year you have approval for the Sarepta, the AAV9 based microdystrophin, which has been approved, which is Elevidys, Relandistrone, moxoparvoic acid, which has just recently been FDA approved for, which is an AAV9 based microdystrophin for Duchenne muscle dystrophy as well, which is equally exorbitant for us. So we need more study to going on.
So that's
Mahesh, you want to ask something, please, Mahesh?
Dr. Pray, I wanted to know which neurological disease you are working on now. Like
no, we are our lab is mainly focused on hematological disease. So
Okay.
we have some interest in lysosomal storage disease, which is Gaucher's disease, but
Okay.
that also has a neuronal component. Okay,
Mhm.
because in Gaucher's disease, there's GBA mutation, which is the L44P, which is the most common Indian mutation. So if the patients, there are reports that the L44P heterozygous individuals may develop a Parkinson's disease at the later stage, right? So we are also studying in that. So we are doing some work on that aspect as well. Yes. So that is which is related to the
Yeah.
neurological aspect.
Thank you.
So I just want to add one more thing. So since the cost aspect has come, because all these therapies are almost like more than $1 million, $1.62 million. So one approach is so making off the shelf approach. Okay. So since iPSC based approach is already approved, FDA approved, so people can think about making off the shelf. Okay, so you can mutate the MHC class 1 and MHC class 2 molecules so that you can use this for many patients so that you can bring down the cost. So that is one approach. It's been applied in other areas, but I'm not sure about the neurons, but people are working on off-the-shelf therapies for many diseases, including CAR T cells. So this also can be used here if you can able to make MHC class 1, class 2 knockout. So then it can also be used for, we can bring down the cost very much.
Dr. Vinod, you've answered something on the, can you answer that live? Because this part of the question was because of your talk, which was there last time, right? Which you've answered, but can you just read out to some people would be wanting to know? Yeah.
Yeah, so there was a question which was, are trinucleotide repeat changes in mitochondrial genome included in whole genome sequencing? And the answer is yes, both mitochondria and repeats are included in WGS, but the analysis is not routine. You require specialized knowledge base and skill sets, and of course, computational tools to do that. There is one question that has come, which is of interest to Dr. Shiva Prakash. Which is from Ankti Sabharwal. Did you observe any bystander or non-specific editing or any other side effects of overexpressing fetal hemoglobin indirectly via gene editing?
Yeah, so we have done off-target cleavage analysis for top 15 genes for both SgRNAs, the left SgRNAs and right SgRNAs by Sanger sequencing, but we didn't see any off-target cleavage effect. And then at the in vitro and in vivo, we didn't see any toxicity or any abnormal growth or anything in our edited cells. So in vitro and in vivo, we didn't see any toxicity or any other abnormal effect. So it seems to be fine.
Yes, so there is one more question. Hydroxyurea also increases HbF. Is genome editing more effective in increasing HbF in sickle cell and thalassemia?
Yeah, so hydroxyurea works only for the sickle cell patients. And it also doesn't help for all the sickle cell patient. Okay, so that is one important thing. So it works only for certain patients, almost like 30 to 40% of the patient, it doesn't work. And then on top of it, hydroxyurea you need to take throughout your life. Okay, so but this genome editing is a one-time cure. Okay, so if you reactivate the fetal hemoglobin by genome editing, so you will be completely out of the small molecule therapy. You will be completely normal, back to normal. Okay, so that is the major advantage. In the other side, in thalassemia, hydroxyurea doesn't work at all. For some reason, the molecular mechanism is not known. Hydroxyurea doesn't increase the fetal hemoglobin. So, so that's where the genome editing makes the mediated HbF will help in that case.
There's a question from Dr. Geeta. Phenomenal work. Congratulations to the team. Would you be able to use this technique in children with X-linked gamma globulinemia?
Yes. Yeah, it is possible. Yes, very much possible. But we are not working on it. So hope somebody can take it forward, these things with the appropriate support, funding support. Yes.
There's one more question. Does it affect 2,3-BPG? Did you consider to study this molecule?
BP. No, I didn't understand the question. What we know the 2BP?
G.
BPG.
2-3, it's 2-3 BPG.
Okay, I'm not sure what is the question.
Talking about 2,3-bis phosphoglyceric acid, probably. I'm not sure.
Okay.
Or if the person could detail the question, that would be great.
Yeah.
Yeah, 2,3-bis
yeah.
phosphoglycerate. That's right.
Okay. I'm not sure about that. I don't know.
Yeah, the Hilda is on as a panelist. I think you can unmute and ask the question, please, Hilda.
Yes, ma'am. Yeah, I was just talking with reference to 2,3-bisphosphoglycerate because normally the action of HPF with the 2,3-bispho is decreased. So I just wanted to know what's the relation with it. Did you check the relationship with it while you did your study? 2,3-bisphospho.
No, madam. No. Sorry, we didn't do anything. We didn't use that molecule. No. Yeah, in fact, we are not using hydroxyurea also because this is totally a genome editing based approach. So we compare it with our unedited and edited. So, but in other studies, we have used hydroxyurea.
Pragita, you wanted to say something or ask something?
No, madam, not at all.
It's
I
okay.
just want to appreciate this wonderful work
Okay.
that has been done.
Yeah.
That's all.
Thank you, madam. Thank you.
Thanks. Harsh? Dr. Harsh?
Yes, ma'am.
Any questions and queries or
No,
comments?
no, ma'am. Yes. Yeah, ma'am.
Okay, fine. There is Dr. Ratna, you can you make Dr. Ratna the panelist? She can speak. She's
Yes,
saying,
ma'am.
yeah. Dr. Ratna, yeah. Is Dr. Ratna, please.
Okay. Hi, Dr. Siva. That was
Hello.
really good work.
Thank you, madam.
Thank you so much. I just wanted to say that I'd like to use it in sickle cell for the non-transfusion dependent, which in the olden times we used to call thalassemia intermedia. So
Yeah.
most of them are intermedia because of slightly higher levels of fetal hemoglobin because of that upstream XMN polymorphism. So those are some patients where hydroxyurea can be used, but definitely not in the transfusion dependent, which is the common of phenotype. That's the common of phenotype.
Right. Yeah. I
Dr.
completely
Ratna,
agree. Yes.
Dr. Ratna, you can also reply to this question of genome editing in inborn errors of metabolism.
Actually, we can edit anything anywhere.
Yeah, I understand.
But
No.
there are so many complexities. It's not so easy. I just feel that slowly as we now get all this, we understand that more than just that one change that we think causes the disease, there's so much of other regulatory elements which we don't know so much about. That's why all these therapies have taken years and years to come. I think people have been working in thalassemia for years, but slowly as the tools become better and everything becomes
Yeah.
better. So
Right.
it's I think just like what Siva said, this is now the time for everybody to take anything can be edited, but you have to work in depth in one field. So like their lab works in hematological disorders, somebody will have to take up different disorders like that.
Yeah.
Because there's just too much of regulation for
Yeah,
every
So I
gene
just want
action.
to add the hemoglobin mutation was identified way back in 1948. It's been so
know.
many years now. It's been 80 years almost, I think. So still we don't have a cure for this disease. So yeah, so now at least we have a technologies coming up. So there is a lot of hope for these patients. Yes.
Yeah, but Shiva, the other question is somebody has already asked about the inborn errors. I asked Dr. Ratna, but for the other neurodevelopmental disorders, there's so much complexities in our understanding about the conditions. For example, autism. You have
Right.
genetic factors, but there are so many. You don't have those exactly. You don't have any set which will rule out. Then you have environmental factors, and then you have the epigenetic ones.
Right.
And that is what I feel. I mean, in this situation, if when you have only when you know the exact genetic, then only you can do the genome editing, right?
Right. Yes, madam.
But I think for conditions like NDDs, which the question has been asked in chat, that I mean, I think it is important for us to focus on the epigenetic factors, because I think those environments, what factors which are going to affect and modulate the gene expression, maybe some good intervention targets, I feel. I mean, for us to act, for any kind of a condition. What do you, think about that?
Yes, madam, that is possible actually. So this is not only used for application oriented. So you can also say example, if the gene is mutated, so environmental factors is others are involved. So we can do some basics research, basic studies in a cell line model, creating a mutation, and then maybe use the FDA approved molecules, screen those molecules using those mutations, and then identify a molecule which helps in reverting the disease phenotype. So in those approaches also, this genome editing can be used, because not only it has to be used for genetic correction for the patients, but this also can be used for basic research aspects. For example, if you're identifying some mutation which is not possible in the patients, but you can create those mutations in the cell line, and then create a model, and then use the FDA approved molecules or novel therapeutic molecules to reverse the disease phenotype. So that is also another approach. People are been using it to get the novel molecules, so that you can speed up the process. Now using these technologies, you can achieve the things much faster than before.
Absolutely.
Yeah, Shiva. So I think I have two questions just to also give a broad overview for the clinicians out here.
Sure.
The first question is, of course, we discussed about how to go about editing. A more important question is when to go about editing. Should we do it as a child, as a fetus, as an embryo, or probably as a gamete, right?
Right.
Of course, there are controversies around this, and at when and where should you act upon, who should be allowed to do so, etc., etc. So that's one question. Or in what, in a sense, what is the future of the technology in terms of when should we do this? Because each of them have their own advantages and disadvantages. And the second more pertinent question is, as pediatricians, pediatric neurologists, who are also training in the field, who are going to see this over and over in probably a better pace than what you see today. Today is mostly in the R&D labs. What should they learn as skill sets going forward to cope with this?
Okay. So yeah, thanks, for those fantastic questions. So the first question, that's a valid question. So so it depends on the disease. Okay. So some disease where the onset is. So if the onset of the disease is very early, it's say example in the birth itself, we have to do embryo editing. So that you can, at least when baby is born, so you will have the normal gene, so that you will not have any symptoms. But some disease, it may the disease onset is maybe after 10 years or after 20 years. So you have a lot of time window is there. So that case, after a birth, then you can take out the cells, or you can do the in vivo genome editing. So it's all depends on the onset of the disease. Okay, so it's an example in the case of sickle cell and thalassemia, as soon as possible, because the fetal hemoglobin expresses up to maybe a year after the birth. So if you can able to achieve the genetic correction within that 1 year period of time, 1 or 2 early pediatric age, so then we can able to get it. So, but some of the other diseases which the onset is very late, right? Maybe in the muscular dystrophy and other things, we may not able to see the symptoms very early. But even in that case, we shouldn't wait till the late stage. So we can able to, if we can able to do at the intermediate stage itself, then we can able to rescue the patient, and the patient can back to normal healthy life. So that is one thing. The second thing, the clinicians, what they have to learn, because these things is going to come, because it's maybe in another 10 years or 15 years of the period of time, these small molecules, the small molecules and other things may go down, I think. So it's completely going to replace, but only the cost factor is the major concern for the developing countries like us. How much you can bring down the cost, because these things, the editing has to be done. Say, if example, if it has to be done at the GMP facility, still it costs more than, it is not going to be like a small molecule based therapy, but at least it will cost maybe few lakhs. Even if it is not going to be a millions, it will cost around couple of lakh rupees. It can be done. So the clinicians, they have to maybe they have to attend more seminars and understand the technology, how it has been used in other diseases. Maybe they have to constant touch with the basic research scientist. So it is like a bridge between basic research scientists and the clinicians. So then only we can able to achieve these things. So when I was starting my lab, I was cleanly looking for the clinicians' collaboration, because even though I know the technology, how it works, and I have done some work, but without the clinical collaboration, we can't able to reach the patient, right? So so the clinicians also has to have a close collaboration with the basic research scientists. So it has to be done with both level. So with the clinical collaboration and the basic research scientists. So they take care about the laboratory part, and then subsequently it will be done to the patient's level. So that is how we say. Best example is I can give from my own experience. So for our sickle cell and thalassemia, the clinicians who are all doing the bone marrow transplantation can able to do the genome editing very well. Okay, so it is the same, almost the same thing. So so you take out, do the apheresis, which is the same in during the bone marrow transplantation as well. So there they are taking the healthy donors cells by giving the busulfan, by giving the GM-CSF or Plerixafor, take out the CD34 cells, and then injecting into the sickle cell patient. So in this case, in our case, so they are taking out the patient's own cells, giving it to us. So we are doing the modification at the laboratory level, and then eventually we will give it to them back. So they will be injecting back into the patients. So this is like the clinician and the basic research scientists, they have to come together, and then with that only it can be achievable. So that is very, very important. So they have to have a constant interaction. So the clinicians also has to have a constant interaction with basic research scientists, and basic research scientist also has to have what is happening in the clinical side. So then only we can able to do achieve these things. So that is very, very important. So maybe the clinicians can also spend maybe 1 or 2 days in a month in the lab, so that they can also see what is this gene editing, how this works in the cells, at least maybe once a month or once in 2 months or 3 months, so they can able to understand better. So at least they should have some interaction with the basic research scientists. So it's on.
Shiva, vice versa. You please
Yes,
come to
madam.
my OPD too. I think would love to have you
Sure.
there. And no, it should be. No, I'm just saying, and we have to
Right.
have more meetings and
Yes.
discussions that is there.
Yeah.
Vinod, you want to add something before I have one question?
Yeah, please go ahead.
Actually, this is not related to genome editing, but this is in the recent advances for management of genetic diseases. Where do we stand today? And in terms of pre-implantation genetic testing and diagnosis, because that is something important. And I don't think we are talking about the advances. I understand that for India, according to what I remember, is just available for thalassemia as of now. For thalassemia, you have a pre-implantation genetic testing and diagnosis available. And obviously, it's a very long shot, very long away for autism and all that. But where do we stand today? And where do you hope, Dr. Shiva and Dr. Vinod, when will it come? And in the end, when are we going to have your whole genome iCards? That we have Vinod to answer, yeah.
Maybe both the questions, Vinod is the right expert
No,
to
sure, you can
answer.
answer that. Yes, I think pre-implantation genetic diagnosis is something that we need to, as a community, as a clinician community, start looking at very actively and closely. One, because it is a field that has boomed really out of control in many ways, and in numbers which are, I would say, staggeringly large in the last 5 years. And in many ways, there is no regulation in what is being tested, how it is being tested, who is being tested, and for what reason is it being tested. And there are a variety of genetic tests, some of them, of course, which are evidence-based, like, for example, thalassemia or sickle cell anemia. Some of them have absolutely no evidence base whatsoever, but nevertheless being tested and nevertheless being marketed. So my take on this is, at least as a community, we need to get together to first lay the rules of the game, because without the rules, you cannot really say anything. In the terms of technology, I think in a few months, whole genome sequencing is probably going to be the mainstay in pre-implantation genetic diagnostics, just because of the fact that it is becoming cheaper. Second, it requires much lesser amount of DNA. And third, and most importantly, that you can do it really, really fast than any other technology available today. So that's my take on it.
Dr. Ratna, would you like to say, as a clinical geneticist, where are we standing? Because as you exploitation to ho raha hai, in many places.
So
where,
that's
what do you
what I
recommend?
really agree with. Yeah, I agree with what Dr. Vinod said. So some indications are very good, but so how would I get it done? I always believe, even today, see, whenever you're doing a pre-implantation genetic testing, you are taking a cell, you are intervening. Any form of intervention in a growing embryo, you don't know what the long-term impact can be. So in my head, it should be very specific. If you have a lady, and there are repeated affected, no? Because in every pregnancy for recessive disorders, we have a 25% risk of recurrence. And we do see sometimes that 1 pregnancy, 2 pregnancy, 3 pregnancies, they are coming affected all the time. Well, yes, then you can offer pre-implantation genetic testing. Within thalassemia, because that's our discussion, I think we have no regulations on HLA testing. No regulations at all. And that really troubles, because you can have a fetus who's not affected with thalassemia, but because he or she is not HLA matched with the child who has the disease, what happens is that they would discontinue the pregnancy, or they do not implant that embryo. So these are really ethical questions, and we don't have guidelines, because all the guidelines that are there for PGT are related to PGT-A, because it's it's for sexing. You should not determine the sex, but I think we all need to look beyond that also. It's very relevant. One more thing which I wanted to add, because we've been having so much of discussion on when are we going to do treatment? Is it in the embryonic stage, at birth? Because this is going to be an exciting era. It's an era we've never seen. And I just want to tell everybody that for infantile Pompe disease, there has been a study that was published in December 2022 in NEJM, where in the group at Duke and at UCSC, the patient was from Canada, and Duke is the hub of treatment for Pompe, and UCSC was the fetal specialist who started intrauterine transfusion with the enzyme. Because normally when these babies are born, already the muscles are affected. And because the glycogen is stored and the muscle fiber rupture takes place, it doesn't help so much, even if you do newborn screening. There's 100% newborn screening for Pompe in the US. So I thought we're actually reaching that era. So this child was to a Pakistani family in Canada who had 2-3 affected. And when they did a prenatal, they opted to continue. And when they opted to continue the pregnancy, that's when this whole concept came up. So these are one-odd cases, but I think that they teach us many things and they help us to think in a slightly different manner.
Thank you. So if there are no more questions, one final message from all the panelists. Dr. Shiva gets the first chance because you're the main speaker.
No, thank you, madam. Thank you for the opportunity. It was very fantastic interaction. Thank you.
In one message, take home message.
Oh, message, take home message.
It's like the
Okay.
rapid fire kind of thing, but I don't have a basket to give, so, but.
No, I think genome editing and the cell and gene therapy is the next big thing in the clinical aspect. So, so it's going to change the field actually. So I could be able to see in the last couple of years, there are a lot of advancements happening. First thing is mainly on the hematological disease. So eventually it will come into other disease as well, because the delivery is the main hurdle for these solid organs, especially the neurons and other things. But that is catching up. So maybe in another couple of years, we could see a lot more advancement. But this is going to transform the field, the cell and gene therapy, both in the terms of AAV, lentiviral, as well as the genome editing. But the cost has to come down. That also eventually it will come down, maybe in another 5-10 years. So yeah, so that's my take-home message.
Thank you, Dr. Vinod. You didn't answer my one question. No? When are we going to have our iCards, whole genome iCards?
Probably in a year's time for people who can start to afford it, and probably in a decade's time for probably 20 to 30% of the country.
Affording, how much cost are you looking at? How are you going to charge us? How much?
I am not going to charge.
Yes.
But having said that, the sequencing cost, I'll say qualified, the consumable cost of sequencing is almost almost reached around $200, and it's going to reach $100 probably in a couple of years. So I think that's a point when I think 0.5% of India can start affording to do whole genomes and probably start replacing a sort of a triaged approach to testing when it's already becoming costly and complicated and time consuming. So I think that's a point when I think that genome cards will become a commonplace. Yeah.
Dr. Ratna. Yeah, please. Your take-home message you have not given us. Sorry, sir.
Yeah, my take-home message is very simple. Last 2 weeks, you have heard about sequencing genomes, that's essentially reading genomes. Today, you have heard about editing genomes or how to go about changing what we already have read. And I think as clinicians, you're already on the treadmill, right? Things are moving so fast because clinicians are only started learning how to interpret sequence genomes. And we are already hearing how to now edit and put it back into patients. I think probably what tomorrow we might be thinking about writing new genomes altogether. So clinicians have a lot more depth and breadth to cover in the coming years.
Yeah, and I'll tell all of the colleagues who are there that Dr. Vinod is a physician scientist. So you guys can also go to the other side. Maybe you'd like to go on Shiva's side and Vinod's side. So you're always welcome.
Always welcome. Yes, madam.
Always welcome.
Yeah, I suppose. Yeah, Dr. Ratna.
Okay, so I think this is the era of therapies. And to be able to give therapies to our patient, I think we need to replicate more of what Dr. Vinod, Dr. Shiva, and all, the labs are doing. And I think it's very important, and it really comes to the forefront with collaboration. Otherwise, a lot of work goes on, and I think the clinicians don't know what's happening in our country. And I believe that we can bring it. And I have a feeling that in the next 10 years, we will be translating a lot to our patients in the country.
Actually, I have one question to all of you. We've discussed in the previous webinars also the ethical concerns which come up along with it. You see, sometimes you have paternity issues coming up. Sometimes you have some condition which is there, like you're having a BRCA positive coming in a neonate. What are you going to do about it? I mean, all those things and how to handle, and the paternity issues I myself have handled. I don't know how. It was difficult. So with two affected sibs and you're getting paternity issues, and then you can't tell, and somebody else will tell, how to do it, and keeping social situation in mind. So I think it's a long way to go before, we can do that. Anything you want to suggest to Dr. Ratna, because you must be handling that more, much more than this before Mahesh thinks about his last statement. Yeah.
It was interesting. We had paternity issues when we did many years ago, before the advent of exomes and NGS, even prenatal diagnosis, no? So when you're doing these, to take sample of the parents for maternal cell contamination at that point in time, not only the mother, but that time we used to take the father also. And we identified, and I remember with Dr. Verma, we had a full discussion that what do we do? Are we going to tell the family? Are we going to do? But we don't have consent to do that. So there is a lot in consenting. And I think we have to teach our students that consenting process is very, very important. And we should not take it lightly. We feel our patients don't understand, तो कुछ भी sign कर देंगे, मगर ऐसा नहीं है. They all understand everything. And they will understand and sign, and we won't be aware, and we can get into trouble. So I think we have to be alert to that. In fact, we've even had, they do non-invasive prenatal testing in a pregnancy, and identified, and that was a doctor couple, identified that neither the egg nor the sperm was theirs, whereas it was only supposed to be a sperm donor. So all this the technology is going to bring up, and I think we really have to be alert to that. And for me, I think that consenting is a very, very important process with genomics.
Yeah, thank you. Mahesh?
I feel as a clinician, we need to, we have a lot to do, ma'am. It increases our genome literacy. We need to know most of the genes and the mutations. So it's a tough job for most clinicians, I feel. But we have no other way but to learn it.
Yeah, I think now genetics is going to take over the whole body, I think, and whole body is always there in the whole body. For all systems, we have to, we will be having genomics everywhere. I think that is the future we have, but we have to be very careful and cautious about all these things. And there was one slide Dr. Vinod had shared with me, and I really liked the slide. That was superior doctors prevent the disease. Mediocre doctors treat the disease before evident, and inferior doctors treat the full-blown disease. So, and that was a saying by some pretty Chinese, Huangdi Nai Chang, 2600 BC, first Chinese medical text. So it is there. We are all trying to become superior doctors, and genetics is taking us from there. Thank you everyone for a wonderful session. And next week, probably, we will be launching a platform in which we'll be sharing the reports all for the VUS and having discussions. And thank you so much, Dr. Ratna, for joining in. And we miss Dr. Madhulika because she's not well. And thanks a lot. Thank you, Shiva.
Thank you,
Vinod.
madam. Thank you.
Thanks.
Bye-bye.
Thank
very
you.
much.
Bye.
Bye-bye.
Bye.